Okay, our next, um, we're going to continue with the research plan presentations, and our next presenter is Kristen Essex. She's from the Organization for Autism Research. Kristen, do you want to share your screen?
Yes, thank you. Okay, is everybody seeing the presentation? Perfect.
Yes.
Awesome. Well, thank you so much, everybody. It's a pleasure to be here with you today. I am Kristen Essex, Executive Director of the Organization for Autism Research. I know we're still a little behind on time, so I'll try to get through this quickly. We're going to do a brief overview of OR and what we do, and then I'll go into a little bit about our research priorities and our suggestions for the strategic plan. So OR was founded in 2001 by a group of parents and grandparents of autistic children. Their primary goal was to help the autism journey for the parents and families to come smoother. Our mission then and now is to apply research to the challenges of autism. We break that mission down into 4 program areas shown here: research, information, education, and community impact. What do I do is the fundamental question a parent asks when their child receives an autism diagnosis, or a teacher asks when they have an autistic student in their class, or an employer when they hire an autistic person in their company. Information, providing that quality, evidence-based information is at the heart of OR's mission. OR has distributed more than 1 million family education and health resources. We offer this information via our website and monthly e-newsletter, The Oracle. Our website also includes our Life Journey Through Autism series of 9 guides that answer the "What do I do?" questions on a series of important topics such as safety, autism knowledge, teaching kids with autism, assessment, and special education. These are all available to download or order physical copies for free. The safety guide specifically has been distributed more than 31,500 times since 2015. We also have resources for siblings and a very comprehensive online sex ed resource for autistic people, which has had more than 87,000 users since June of 2023. Within the online sex ed guide, we have a safer dating curriculum and mental health guide for college students. Finally, we offer the only resources expressly designed for military families impacted by autism, supporting more than 95,000 military families since April 2023. Education is another mission area. We offer free webinars that seek to deliver targeted information on topics of high interest to the autism community. We typically hold 6 to 10 webinars a year, led by autism experts from around the country. Topics have included transition planning, employment, dating and relationships, mental health, healthcare, and many more. In direct support of education of autistic young people, we have a scholarship program that supports students pursuing undergraduate studies, undergoing vocational technical training, or participating in transition life skills courses after high school. We also offer a tech scholarship for autistic job seekers pursuing certification or job skills in a tech field. As community impact signals, the programs here are OR's major initiatives. OR's Kit for Kids peer education program has literally gone around the world, helping neurotypical peers better understand their classmates with autism. We've continued to expand this program with our peer education grants that help teachers and club or event leaders spread autism acceptance through the use of OR's Kit for Kids program. Most recently, we've introduced our friendship tip sheets that promote acceptance and friendship in schools. Higher Autism is our jobs portal that offers resources to help autistic adults find meaningful employment. Initial concept was an online jobs board to connect autistic job seekers with employers willing to hire them. We launched that pilot program in the Northern Virginia area and then expanded nationally in 2020. We have since grown the program to include job search resources and one-on-one resume and interview prep through our Navigator volunteer program. And finally, research. It's part of our name and the foundation of our mission. We encourage and support research through 2 programs: the annual Applied Research Competition. Oops, sorry. Sorry. There we go. The annual Applied Research Competition and the Graduate Research Grants Program. The Applied Research Competition is our major grants program. We fund 6 to 8 studies a year with grants up to $50,000. These are pilot studies that allow researchers to collect data and test hypotheses as an essential stepping stone to more in-depth follow-on studies. We are currently emphasizing grant awards to junior investigators to help build the next generation of autism researchers. Additionally, to support the future stars in autism research, we fund Graduate Research Grants Program. These are small grants to support master's and doctoral students conducting research related to autism in pursuit of their degrees. Since 2002, OR has funded 162 applied research grants and 216 graduate research grants. Those investments have produced at least 186 journal publications, with many appearing in the field's leading peer-reviewed journals. But publications are not the endpoint. The real value is what happens when that research informs practice, leads to larger studies, and becomes part of sustained programs of research. OR's research portfolio has translated into effective interventions and aims to strengthen the systems, services, supports, and communities that improve the overall well-being, health, and quality of life outcomes for autistic individuals and their families across the lifespan. Driven by community priorities, we advance dignity, respect, quality of life, and individualized support across the full spectrum of needs. The impact of OR's research investments extends well beyond the initial grant period. Many OR-funded projects have developed into sustained programs of research, generated multiple publications, attracted larger federal and foundation investments, and helped informed interventions now used in clinical, educational, and community settings. Examples include PEERS, JASPER, Unstuck and On Target, parent-mediated interventions, and transition supports for autistic adolescents and adults. This is why pilot funding matters. A relatively small early investment can provide the evidence needed to launch a much larger body of work. This chart illustrates the growing demand for applied autism research funding. For the 2026 grant cycle, OR received more than 300 letters of intent, far more than at any other point in our program history. Yet our resources allow us to fund only 6 to 8 applied research projects each year. This gap represents a significant amount of promising research that may not move forward or may be delayed while investigators seek other funding. It also demonstrates how much interest exists in addressing these practical questions facing autistic people, families, school, providers, and communities. As the strategic plan is developed, we believe it is important to consider not only which areas of research should be prioritized, but also whether sufficient research funding pipelines exist to develop promising ideas, test them in real-world settings, and position them for larger investment. These 4 studies illustrate what OR means by applied research. The goal is not simply to describe autism, it is to develop and test practical approaches that can improve daily life. Facing Your Fears adapted a family-focused anxiety intervention for teens with autism and intellectual disability, a population that is often underserved by community mental health providers. The study focused not only on anxiety, but also on their participation, social experiences, family functioning, and quality of life. The PEERS virtual coach explored whether widely available technology could help teens practice social skills more naturally without relying on a parent or teacher to coach every interaction. The Joint Attention Study tested a social skills intervention that teaches joint attention, showing, pointing, and sharing attention among young nonverbal autistic children, a skill that is closely tied to later communication development. And our current assent-based ABA study brings autistic and neurodivergent community members into the development of a more affirming approach. It asks how providers can recognize both assent and withdrawal of assent, particularly for people who require greater support. Together, these studies reflect the kind of research we hope the strategic plan will elevate it: practical, inclusive, community-informed, and connected to outcomes that matter across the spectrum in everyday life. These are the areas OR believes deserve sustained attention. They cover the lifespan and reflect concerns we hear repeatedly from autistic people, families, practitioners, and community partners. Several themes cut across the list. We need stronger research on communication and belonging, on community living and the supports that make participation possible, and on physical and mental health across adulthood, including midlife and aging. We also need research that is more inclusive of people whose experiences have historically been overlooked, including people with intellectual disability, non-speaking autistic people, people with high support needs, and people whose experiences are shaped by race, gender, geography, income, and other intersecting identities. Gender, reproductive, and sexual health should be treated as essential health research, not as a peripheral topic. Care should be inclusive and affirming while also addressing safety, access, and informed decision-making. Most importantly, we should not study these areas in isolation from the systems people must navigate. The effectiveness of an intervention matters, but so do availability, affordability, workforce capacity, cultural responsiveness, and whether it can actually be used in real-world settings. These priorities lead to several questions that we believe should help shape the strategic plan. Research does not exist in isolation. Ultimately, it should improve people's lives. Seeking answers to these questions can help bridge the gap between research and meaningful impact. First, how do we move promising work beyond small pilot studies? OR is proud to fund stepping stone studies, but the field needs stronger pathways and resources for replication, implementation, dissemination, and long-term sustainability. Research should tell us not only whether an approach can work, but what it takes to make it work in schools, clinics, workplaces, homes, and community organizations. Second, what affects access to services and who is most likely to be left without them? Medicaid and CMS requirements, eligibility rules, reimbursement structures, workforce shortages, geography, language, and socioeconomic barriers can determine which services exist and who receives them. These barriers may be especially significant for racial and ethnic minorities, rural communities, people with profound autism or other high support needs, and others who have historically been underrepresented in research. We need research that connects policy and system decisions to service access, continuity, equity, and real-life outcomes. Third, we need solutions to persistent capacity gaps, including long residential wait lists, limited mental health expertise, and shortages of appropriate providers and direct support professionals. Describing these shortages is not enough. Research should test practical, system-level responses and identify what is needed to sustain them. We also need to consider how the field responds to controversial, unproven, or scientifically discredited treatments. Families are often navigating complex needs, long wait lists, limited services, and an overwhelming amount of conflicting information. Simply dismissing their concerns is unlikely to build trust or change decisions. We need research-informed communication strategies that clearly explain the evidence, identify potential risks, address misinformation, and help families evaluate treatment claims while engaging them with empathy and respect. Gender, reproductive, and sexual health also deserves greater attention as essential components of health and quality of life. Research should help ensure that care is accessible, affirming, developmentally appropriate, and responsive to the diverse identities and support needs of autistic people. It should also address safety, informed decision-making, provider training, and the barriers that prevent people from receiving appropriate care. Finally, the field should be explicit about the outcomes it values. Success cannot be defined only by symptom reduction, compliance, which is not the same as true assent, or conventional measures of independence. It should include autonomy, dignity, communication, belonging, safety, meaningful relationships, community participation, and quality of life as in collaboration with autistic people, including those who communicate differently or require lifelong support. We recognize that I-ACC's I-ACC role is to guide and coordinate research priorities, not to implement services directly. Our recommendation is that the strategic plan support research examining how effective approaches can be translated, adapted, and sustained in real-world settings. That will require coordination with federal agencies, service system, practitioners, autistic people, and families. Our core recommendation is to pair scientific rigor with lived experience and to treat implementation and meaningful outcomes as research questions in their own right. By supporting the evidence needed to move effective approaches into practice, I-ACC can help ensure that research investments ultimately produce meaningful change. Thank you.
Thank you, Kristen. Any quick questions for Kristen? Okay, if you could stop sharing your screen, I will try to. Our next presenter, or did someone have their hand raised? I'm sorry. No, okay. Our next presenter was supposed to be Dan Geschwin, but Dan was unable to join us, but he sent me his slides. And for the sake of getting them just into the public record, I'm just going to go through them really quickly. But let me say very publicly, I am no Dan Geschwin. I am not a geneticist. Um, let's see if I can get them up here. Okay, so I think what Dan wanted to share with us is very similar. I think it builds on what Manny discussed with regards to um, many genetic causes, but understand using that to really drill down towards the biology. And I think whereas Manny focused more on animal models, Dan is focusing more on organoid models. But he sent me just three quick slides that as I said, we have 100 genes now that are linked to autism spectrum disorder, and that each of these genes has a different story, but we see the same disrupted pathways and the same disrupted brain patterns again and again. So, can we use these genes to really drill down on understanding biological targets? He is focused on using iPSCs to develop organoids. I think this is a slide that talks about the process of developing organoids and then speaks to the value of organoid models to recapitulate human fetal development. And he also, just as Manny started to outline the two projects in this space, ARIA and the one at NIH. I think there's well, we can get to that later. And so I think in his lab, he's working on developing these organoid models and at the same time working on a parallel study using CRISPR technology. But the so what of this approach is, as I said, that the iPSC models really are, the science is showing that they really do very faithfully capture human fetal cortical development, which, as we've talked about, is the peak period where genes are affecting the development of the brain. He's talking about the importance of scale, which Ekemini already spoke to as well. And I think he will probably, when he joins us for our next meeting and can do this on his own, share a little bit more than Manny did about the NIH Gene Consortium, which is generating more phenotyping data with and tying that back to organoid and CRISPR models. So if there's anyone on, do we have geneticists? I don't think that. If anyone wants to add to this or is more familiar with Dan's work, I just want to make sure that it was available for the public to see so that it can be included in the plan if the group decides to. Okay, well, in
Allison,
the
maybe
interest,
I'll just say one quick
thanks.
thing about this is that almost everything this field has talked about with respect to genetics is germline genetics. It's what you get out of a lymphocyte in the peripheral blood, and that's where you measure it. There's every reason to think that many neurodevelopmental disorders are actually due not to a germline mutation, but to a mutation that occurs specifically in certain cells in the developing brain, where you have very, very rapid reproductive cycles, and you just have errors that get generated in brain development. And to do that, you actually have to do the genomics on those cells or in that particular brain area. So it requires brain tissue to be able to analyze that. And Dan's lab is one of the very few that has done that, has begun to actually generate maps of gene expression in the very specific brain areas that have been associated with autism. And I think what we'll discover over time is that those mosaic or those cell-specific DNA changes may turn out to be incredibly informative, even when you don't have a germline signal that goes with them. So that's a whole area that's got huge potential, and Dan's lab, along with a few others, have really been at the forefront. Kelsey could probably say more about that
No,
than I
I would
can.
just add Chris Walsh to that list of people who are looking at postmortem brain tissue, looking at mosaic, the role of genetic mosaicism.
Right, exactly. So this is like a different way of thinking about genetics. It's not really saying, oh, every cell in the body is going to be affected, but that is a group of cells in the brain. And actually, this is the history of cancer genomics. So when you look at cancer therapeutics, it's not coming from germline mutations. It's mutations that are specific to the tumor. That's where we really understood causal mutations and we were able to develop the interventions that would make a difference.
Okay. Thank you, Dan. I will stop sharing. Our next presenter is John Haggerty. John, if you're speaking, we can't hear you. I can't hear you.
All right, there we go. Is that,
There you
can you
go.
hear me? Okay.
Yes, yes.
Sorry, yes, just trying to get everything up. So, yeah, so I think we've had a lot of discussion today on the biological side in terms of especially some of the more basic research going on. But, I think it's also very important to discuss the translational research and really priorities to help advance precision therapeutics and supports for autism. There we go. So just a little bit of background. So my training is in primarily cognitive behavioral neuroscience, and I've been focusing on developmental disabilities, especially autism, over the past 10 years, and using a number of non-invasive neuroimaging approaches, but in conjunction with clinical research studies and clinical trials that we're conducting. And so we have a number of different areas that we're targeting this at the Stanford Autism Center. Just for my disclosures, I have federal, institutional, and several nonprofit research funding and do some consultation for those nonprofits. But I think it's also important to help characterize that in addition to studying autism, I'm also neurodiverse myself and have personally benefited from access to a number of these psychiatric supports and psychological therapies that are under investigation. My wife is a speech and language pathologist that works primarily with autistic children with communication delays, and we're both primary caregivers for an autistic child. So these are areas that we are very actively thinking about on a regular basis. And so, through the research as well as our exposure to members of the autism community, it's very clear, and I know I don't need to spend too much time getting into really how variable the presentation is in this population. And this is outside of even the core domains themselves that can be very variable. And so it should come as no surprise that really we're talking about very variable neurobiology, and really the existence of neurobiological subtypes and subgroups within the spectrum. And this is something that on the translational and clinical research side, when it comes to characterizing the neurobiology of autism, we run into severe limitations in terms of the traditional case control comparisons that research has relied on. Because if we have these subtypes comprising the population, where there might be individuals with more similar cognitive behavioral profiles that are shared or biological profiles that are shared, using these traditional approaches will wash out these really relevant signals. And so it really requires this very directed approach of taking subgrouping into account. But this is something that is also further complicated by the fact that we're talking about developmental trajectories where really we're trying to hit a moving target because these relevant mechanisms may also change over time throughout development within an individual. And so some of the areas in clinical research in autism that is really important to directly address in our research design is how are we going to really embrace this variability? And so the first is having a very specific focus on the specific domains or support needs that we're trying to better understand. And this is going to require, as has been mentioned before, some improvement in measures for characterization, but really specifically with a focus on these specific domains themselves and subdomains within those that we're trying to characterize. Because as we're able to characterize those subdomains better, we'll be able to then take that information to help with the identification of not just this neurobiology underlying autism, but really the neurobiologies underlying these different areas that we're trying to build up. These will then help us with the identification of biomarkers that can then help with this subgrouping to then really inform these biologically based intervention approaches. And then as we test these approaches, it'll be important to really adapt our clinical trial design to focus on these subgroups, as well as incorporate more real world interactions between the types of therapies that are being applied. So, for instance, between the outcomes of any pharmacological therapies, for instance, on how well and efficacious individual cognitive behavioral therapies might then be. And this is an area where we can see a little bit of a roadmap in terms of some of the developments that have been done in adults. So here, for instance, looking at recent progress in mood and anxiety disorders, we've seen the beginning of some types of neurosubtyping, for instance, that can help differentiate some of these subgroups. So similarly, if we're looking at patients with these umbrella diagnostic labels, we may be able to then parse out some of the variability that's going to define their more specific biological subtype. And then once we can identify those, these will then help identify potential therapeutic interventions that may be more appropriate and more beneficial for those individuals. These measures that help with the characterization can then also potentially provide early signals of clinical impacts that might occur before observable changes in cognition and behavior. And so the application of these may really help deal with one of these big limitations in autism research being these extreme individual differences that we need to be able to account for. And there's a number of different ways that we can approach this, but one specific area that we've seen some developments in the same kind of mood disorder and depression group, and significant interest in autism for this precision therapeutics is transcranial magnetic stimulation. So this is a non-invasive brain stimulation technique. Essentially, there's a magnetic coil that's placed above the scalp, and it will induce electrical changes in the brain. And this is thought to have some effects on neuronal activation and neurotransmitter release, but really that it may be able to help support neuroplasticity. And it's been cleared so far for things like depression and OCD. It's now been cleared for adolescents as well in those groups, with interest for pediatric populations because it's typically very safe and well tolerated in most individuals. But what's really important for the development and future of this line of investigation is that we're starting for these individuals to see in cases where the depressed patients are not responding to more standard of care treatments, they're starting to see more insurance coverage for these types of therapies, as well as more infrastructure that's been growing at an annual rate. There's been interest and some research going on in the autism community for these areas, but until recently, we haven't really had the robust clinical trial data to really support the potential efficacy of these approaches. But for instance, just last month, there was a large multi-site trial that was published using a form of TMS called continuous theta burst stimulation in autistic children. And they saw a significantly greater reduction here looking at social communication on the SRS in scores, suggesting significant benefits in these groups. This study was primarily focusing on ways to apply this type of neuromodulation in a very scalable and feasible design. So they were targeting just primary motor cortex, but were establishing that there might be potential benefits from this course and have the ability to then focus on more precise brain regions that may be more efficacious for specific symptom domains or areas for growth that we're trying to target in autism. And with this neuromodulation, we're able to use different types of biomarkers to help increase our precision with being able to target individual areas within the brain that might be relevant. And this is something that, for instance, within patients with depression, they've used things like functional MRI to help identify individual targets within the target circuits that they're trying to get at, where there'll be slight differences for individuals on where might be the best spot to try to target. And recently, there was also a publication starting to quantify how much of a benefit is this actually providing. And so here, for instance, they saw that it was able to increase response rates by about 20% and also increase remission rates going forward. So we can see a combination here where there may be some individuals that kind of standard, more scalable and feasible applications are beneficial, but the more precise neuromodulation may provide even more benefits depending on the targets. And this is an area that we're actively studying at the moment. So for instance, right now we're conducting a clinical trial where we're comparing two types of theta burst stimulation. One that's supposed to kind of more generally thought to stimulate the circuit, and one that are supposed to potentially more inhibit the circuit. And we're looking at outcomes in restricted and repetitive behaviors, especially compulsive behaviors, insistence on sameness. And so far, we've been seeing very promising results that are actually supporting the potential for theta burst stimulation to be used. And this is including in adults with profound autism. But this is not just limited to these neuromodulation techniques. This type of precision therapeutics are being studied in terms of pharmacological interventions. So here, able to better predict who's going to respond to a drug like memantine based on MR spectroscopy measures of neurochemical concentrations in the brain. And then here's some of our work focusing on looking at brain structures within language regions in minimally or nonverbal or non-speaking children that are autistic, and showing significant relationships and prediction of how much they're benefiting from those types of pivotal response treatment therapies. But this area is really kind of brand new and has a long way to go in terms of us really being able to understand the impact and utility of this. So, for instance, right now we're in the process of conducting a literature search, and we're looking across all clinical trials that have been done in autistic patients that have incorporated any of these non-invasive neuroimaging measures at baseline and then reported on their ability to predict the outcomes from treatment. We started with about 4,500 articles, but after looking through the criteria across the entire array of different clinical trials and therapies that have been conducted, we have found 23 studies so far that actually have reported on the potential for those biomarkers to then predict outcomes. So it's very promising, but a long way to go. And so really where we see this going and where we hope the research is focusing is that if we have, an autistic person that's seeking some sort of support for areas that are affecting their quality of life, we want to be able to use these biomarkers to help characterize their individual neurobiology. So we can then use that to inform the treatment approaches and support plans across whatever those modalities might be. And then hopefully this person will then develop new skills or experiences some aspect of improved quality of life. But this involves an array of research where we need studies to really identify the genetic environmental interactions that are occurring during neurodevelopment. So, still supporting the basic research going on, but we're also looking at, for instance, twin models in the brain in autistic twins and trying to identify impacts on different brain structures. Studies to really determine the neurobiology underlying specific areas that we're trying to target so that we can then classify subgroups based not solely on brain or behavior, but really the interaction between the two. And then these will allow us to develop those biomarkers to help hopefully get autistic individuals seeking support into the right treatments more quickly. And to then studies to then implement interventions that are going to be informed by this biology. So we still have a long way to go in this regard. So ultimately, for these research priorities, I think the most important is that we just need to embrace this variability. So what factors are mediating these individual differences that are going to be clinically informative? How can we characterize and focus on these subgroups, utilizing more specific and objective measures so that we can tailor clinical trials, which is going to be very important, to specific subgroups themselves. And this is going to be something that will hopefully help us get over this barrier where we keep getting washout effects in our clinical trials, where some participants are responding very well, but others are not. This is going to be especially important to continue across the lifespan, where these biomarkers and mechanisms we're targeting can significantly change. And to also examine interactions between therapies and combinations of therapies in, for instance, how something like TMS could potentially help support neuroplasticity, which then may help increase response to cognitive behavioral therapies. But finally, I just also want to emphasize that as we go in this direction for trying to identify subgroups, it's also going to be very important that we be mindful that we continue to advocate for access to care and support services for all autistic people, regardless of what subgroups we might identify, so that we can continue to get everyone the type of support that they really need. So I want to thank our team at the Stanford Autism Center, our funders for this research, and really thank you for all of the families that have done this type of research with us. I know that it can be very intimidating and daunting to think about doing some of these neuroimaging approaches, but I can say that we have built approaches that can support people from across the spectrum, from the profound all the way through the rest, to be able to go through these types of procedures, and that this type of data is critical for advancing this area of research.
Thank you, John. So unless there's an urgent question, in the interest of time, I'm going to ask, does anyone have an urgent question they feel they just can't hold it in? Okay, thank you. That was super interesting. We're going to move on to Dr. Eric Morrow.
All right, I have to unmute. Can people hear me now? Great, thank you. So thanks, John. John set up my talk, I think. So thank you very much. People can hear me? Thumbs up. Good. So what I'm going to talk about today, I was asked to speak to, I think, a critical area for the strategic plan. And I've put this up here, autism subtypes. And I think John's talk really led into this. And I'm going to present a little bit of background on who I am since I'm new to the group. And thank you very much for including me. It's really an honor to be included. And I'm going to present this in the context of a new, very, very exciting Autism Institute, the Lurie Autism Institute, where I'll be moving to be the founding director. Really humbled by this opportunity for me. I think it's a career opportunity. And I think for the field of autism, this is really a really important institute and moment. So thank you for this opportunity to present today. So by way of my background, I'm trained as a physician scientist, so I'm a psychiatrist by training. I'm also a basic scientist, geneticist, and molecular neuroscientist. I was trained in Boston, and then in 2009, I moved to be a faculty member in an interdisciplinary setting in the Brown University Brain Sciences Institute. One of the things in my talk today I'd really like to stress is the importance of interdisciplinarity in autism research and innovation. And part of the things that drew me to Brown now more than 15 years ago was this interdisciplinarity and also a hospital there called the Emma Pendleton Bradley Hospital, which is actually the nation's first freestanding child neuropsychiatric hospital, has a range of clinical services for children with severe behavioral challenges. And I'll tell you in a moment, my area of research is in the area of profound autism. So this access to this hospital really appealed to me. Now, after my career in New England, I'm moving to Philadelphia again with the honor of directing this newly founded Autism Institute that spans across the Children's Hospital of Philadelphia and University of Pennsylvania. So in my research career, I've been devoted to families with rare neurogenetic disorders that includes autism and in particular intellectual disabilities. I've had the privilege of working closely with families around the world who have children with a particular syndrome called Christensen syndrome, and working using genetics to really define the brain mechanism and as it might guide you to molecular therapy. And I've worked a great deal in the profound autism space. I've also had the opportunity of working regionally in my community in Rhode Island with families who have children represented or adults across the autism spectrum. And one aspect of that work is autism in adulthood, and that's been, I think, a major focus of my own work and will be a major focus of the Lurie Autism Institute. What I will say is that I'm deeply committed to interdisciplinary research. I've mentioned this a few times, community engagement, in particular, drawing trainees from diverse fields into the autism realm. So I'm really excited to now be a part of this new Lurie Autism Institute. I'm presenting today before I'm officially the director. I'm officially the director September 1. This institute was founded a little over a year ago from a transformational gift from Jeffrey Lurie and his family, given to both University of Pennsylvania Medical School and the Children's Hospital of Philadelphia. And just so by way of background, CHOP and Penn Medicine have longstanding historical uh, record of collaboration. CHOP, of course, is a major children's hospital, and the Penn Medical School is one of the major medical schools that has been really successful in translational science. Um, so let me get so and so this institute really has the ability to work with basic science, translational science, develop clinical readiness for clinical trials, as well as disseminate these treatments to the community. There's shared genomics, data science, translational therapeutics across these two major institutes. So if you look at some of the numbers that have been shared with me, there really is a very strong track record of moving science to FDA approvals, including in CAR T therapy, mRNA vaccines, and I think in particular in molecular and gene therapies, there's a long-standing and deep experience in molecular and gene therapies in this setting, which I think is ripe for the progress of the last 15 or more years in rare genetic disorders, and which really contribute substantially in the realm of profound autism. There are many, many different grants and already funded faculty that are focused on autism. There are many more who are autism adjacent, and I think part of my role will be to make bridges across disciplines and departments and to help maybe draw some more people who are autism adjacent into the autism space. There's a good infrastructure of patients, both childhood patients at CHOP and then adult patients in a program called the ASPE program at Penn. And there's a very deep biobank and clinical medical record data set, and already a sizable number of genomes sequenced. So for this institute to be founded at a time when there's been large progress in genetics and in artificial intelligence, I think it's a really exciting opportunity. So the a major goal, overriding goal for the Lurie Autism Institute is to really speed interdisciplinary innovation for bold new discoveries and bold new efforts across the autism spectrum and across the lifespan to really serve the global community and to be, a hub for international collaboration, a magnet for trainees internationally, and to capitalize on the strengths of CHOP and Penn. I think that it's very important to really emphasize the importance of basic science and understanding neural mechanisms and developing different biologically based subtypes uh, across the autism spectrum, and that is an emphasis in the Institute. We're working to engage families inclusively across the autism spectrum for research across the lifespan. And the goal is to develop really test big ideas for autism subtyping, care, and treatment. There's a major emphasis in molecular therapies and rare genetic disorders. There's a major emphasis in using artificial intelligence to analyze computer vision methods in autism subtyping and phenotyping, artificial intelligence and drug repurposing. These are all exciting new innovative directions of the Institute. Um, and with the motivation of improving measures of quality of life for individuals with autism, really across the spectrum. what we really are hoping is that this institute will be internationally an epicenter for autism research and training, gives us an opportunity to really draw trainees from diverse specialties to come to a center to train in an interdisciplinary way. And to make this even more exciting, we're collaborating with actively, in addition to this university-based, medical school-based, children's hospital-based Lurie Autism Institute, we're collaborating actively with the Eagles Autism Foundation. It's another major uh, initiative from the Lurie family and the Eagles, and working with the Philadelphia community in this work. So at that point, I'll stop and take questions or comments if there are.
If anyone has a really urgent question, otherwise. Okay, then we'll move on to our last presenter in this section, Dr. Jenny Fan.
Hi,
Thank
all.
you, Eric.
Can you everyone hear me okay?
Yes.
Okay, I'm gonna see if I can share the right thing. Okay. Can people see the presentation or my notes?
Right now, we're seeing your notes.
All right, let me fix that. Try that again. Sorry.
It's okay.
I don't know how to switch it.
I think if you try to take it off presenter mode,
Oh, thank you. I think that's what's going on. Thank you. Uh,
it's a strange world where I give tech advice. Let's just be clear about that.
okay, let me try
Really
again.
log into Zoom.
Yeah. How do I? Oh, okay. I'm so sorry, everyone. It's not letting me share my other screen.
You want to try to email me the slides and I can try to share them, or
Yeah,
do you
let
want
me
to
do
just try
that.
again? Okay.
Thank you, Allison. I appreciate you being flexible. Okay, I just sent it, Allison. Thank you. I was gonna say I will try to be efficient since I'm what stands between everyone and lunch, and that was not efficient.
Okay, so far I have not received it. Okay, we have some tech advice from Matthew Belmonte saying if you are using a Mac, you need to select the appropriate option under the monitor's control panel to show the second screen instead of mirroring the display. I don't even understand that, but if that
I
helps
have a
you, Jenny.
PC, so I
Okay.
think that doesn't apply in my case.
Okay, so I see your slides.
Okay.
I'm going to download them. Can you see them now?
Yes, we can see the slides, if everyone's okay with it being shown this way. Thank you, Allison. So first, I want to thank the committee for allowing me to be a part of this group. I am really honored to be here. I love storytelling, so my presentation is told through that style. This presentation is at the capacity of where I, along with other early career IDD researchers I work with, believe is an underfunded research area. So for you to get to know who you're listening to, I want to share a little bit about who I am while giving context for why transition age youth is a developmental period in need of research, service, and support. Before I was a scientist, I was a mentor. As a psychology undergraduate student in New Orleans, I spent several years as a youth peer mentor, working with young people who were about to age into adulthood without much of a plan for it, which motivated me to study adolescent development in grad school. I wanted to understand individual, family, and environmental effects on adolescent development outcomes and their transition to adulthood. I came to this research because of the people in the community where I served, well, when I served as a parent member on the Iowa Autism Council. A dad who came to a public meeting thought that the council was able to point him to resources on how to support his autistic teenager who started puberty. He also wanted to know how to support his son through the transition from adolescence to adulthood. We didn't have answers for that dad on that day or even days after. So I went to the literature and found almost no research in this area, specifically puberty and autism and related behavioral health outcomes. This was in 2016. I then switched research direction in my PhD program. Next slide, please. So here's the question that has driven my research. Why does adolescence matter or should matter to all of us? Why is this particular developmental window so consequential? My background is in behavioral endocrinology. In my lab, I study how the body's stress and hormone systems develop across adolescence and into young adulthood, and how a young person's environment shapes that neurodevelopment. In a longitudinal study of adolescent twins, my colleagues and I found that family stress and family psychopathology measurably change the way adrenal and gonadal hormones co-regulate as teenagers go through puberty. The takeaway here is simple. The biology of adolescence isn't fixed. It's still being built, and it responds to what's happening around the young person. That study wasn't about autistic teenagers, and it wasn't about the transition to adulthood. It was about adolescent development broadly. What it shows is a mechanism that adolescence is a sensitive period when these systems are still forming and still shaped by environment and support. Autistic teenagers go through the same biological window. Everything that follows rests on that. Next slide, please. I have two autistic sons. One has an intellectual disability and one does not. And the transition to adulthood doesn't look the same for either of them. The harder fight hasn't sorted neatly by diagnosis. The supports fall away on different timelines, in different ways, and not always where a label would predict. I've had to advocate for both of my sons differently, but equally difficult. Here's what gets lost when people talk about transition. A services cliff shows up in specific places. The day an IEP mandated service ends at high school exit with no equivalent waiting on the other side. The handoff from pediatric to adult healthcare, the case manager who disappears the moment a diagnosis stops being the school's legal obligation and becomes the family's problem alone. So this chart is the US Department of Education data on how students actually exited high school in 2019. It's broken out by disability category. Start with autism on the left. 71% graduated with a diploma. Dropout sat at 6 to 8%. On its own, it reads as a success story. Now, look at the intellectual disability category next to it. Diploma completion drops to under half at 48%. Exiting with a certificate instead of a diploma nearly doubles to a third of students. And down at the bottom, reaching maximum age with no diploma at all, that's 5% for intellectual disability compared to 3% for autism. Nearly every measure moves in the same direction. That's what a single word on a policy document can't show us. Autism is a category, as a category, looks like a relative success story next to the broader disability population. The other disabilities column bears that out too. But intellectual disability, whether it stands alone or sits alongside an autism diagnosis, is doing more of the work in predicting outcomes than the word autism is. Support need doesn't sort by label on the file, it sorts by what a young person actually needs, and a diagnosis alone won't really tell us that. Next slide, please. So families named this before researchers did. They called it the services cliff, and the name stuck because it was accurate. Let's look at what we know nationally. Since that's just how numbers are usually tracked and reported, 2 in 3 autistic young adults are not in work or school in the first 2 years after leaving high school. That disconnection doesn't resolve on its own. Follow the same population further out into their early 20s, and 37% are still disconnected, not working, not in school, not in any training program. Now put that next to their peers. Young adults with other disabilities land under 8%, and that's not a modest gap. I want to flag where these numbers come from. They trace back to the 2015 National Autism Indicators Report, built on the National Longitudinal Transition Study data that was collected in 2009. That's over a decade old. A single snapshot from 2009 can't capture what we just saw in the last slide, that autistic young people with and without intellectual disability are facing different odds. If this is still the number driving policy, we're arguing from data that predates the problem it's used to justify. And notice what a top line number like this can't tell us on its own. It's one figure standing in for an entire population. It can't show us how unevenly the cliff falls depending on support need. If we can't agree on to measure this population on a current basis, we're not positioned to catch that unevenness either. Next slide, please. So put the two pieces together. This is a grossly simplistic illustration of neuroplasticity, so please bear with me. And there's also some synchrony between my presentation and John's presentation. So we pull support away at the exact moment the biology is least settled. The services cliff lands right inside the developmental window when stress physiology, neurobiology, and regulatory systems are still forming. We're not just failing these young people on paper, we're pulling, the scaffolding while the biological building is still going up. I can't tell you we've proven the timing of the cliff, that it causes lasting biological harm. Nobody has studied it that way. What I can tell you is that everything we know about adolescent development says this is the wrong moment to withdraw support. That's a hypothesis worth testing, and right now it is a critical gap in the literature. And whatever the cliff does, it almost certainly does it differently depending on a young person's support needs, and that's the part we keep flattening. Next slide, please. So the window is still open and the biology is still forming. That's exactly the kind of question worth studying. But in my field, the approach is usually the same. We find a biological target and then we rush to fix the concern. Oxytocin is the clearest example. It's sometimes called the bonding hormone, and for years, researchers have proposed giving it to autistic people as a drug, on the hypothesis that raising oxytocin increases social engagement and reduces what gets labeled as core autistic behavior. I've pushed back on that with a group of fellow autistic and neurodivergent scientists. Oxytocin was the wrong target for three reasons. First, the people asking for it were usually parents, not autistic people. Second, the outcomes being measured were parent and clinician ratings of social behavior, not anything autistic people said that they wanted as an outcome. And finally, the most rigorous synthesis to date, a pre-registered meta-analysis correcting for publication bias, found only a small effect on social outcomes that shrank further once bias was corrected, and no effect on repetitive behavior at all. None of that means we stop studying biology, because I am trained in biology, so I would not advocate for that. What I mean is that we ask a different question, not how do we move a hormone level, but what actually happens to a developing nervous system when support is there and when it's taken away. That's a question I believe autistic people and parents can agree they want answered. Next slide, please. The ask is pretty simple. Let's put the transition to adulthood into the strategic plan and study it as a developmental window, not as one population falling off one cliff, but as different young people falling at different rates, depending on the support that they need. Research on the biological mechanisms of a sensitive transition period can inform supports and services for young autistic people. I might have been able to, actually, it might help that dad who attended the Iowa Autism Council meeting that I mentioned earlier and might have supported his son's teachers and providers had he known this information. Right now, it's a gap, and it's a gap during the years that matter most to young people and their families. We study early childhood heavily. We are now studying adulthood pretty heavily. And then the years in between, when the biology is least settled and most responsive, are the years we study the least. And they're the years support are removed the fastest. This can have lasting impact into adulthood. I can help design that agenda item. I can bring rigor to the developmental science side, including being honest about where the evidence is strong and where it isn't. And I can help us stay connected to the autistic community and the families living this, so that the questions we ask are the right ones. And I came to this work through supporting marginalized youth in New Orleans, and I come back to it every day as a mom, and I'd be glad to do this work with the committee. And then last slide, please. This is my information. I'm the director of the Bean Lab at George Mason doing this work. And if you would like to contact me and talk more, there's my email. Thank you for listening.
Thank you, Jenny. Anyone have any urgent and pressing questions? Because we are time. Okay, so we'll bring this portion of our meeting to a close. On our agenda, we have our break for lunch. As I said, we're a little bit over time. So what I'm going to say, we do want to be back exactly at 2 o'clock to start public comment. It's important that public comment be on time, because I know people dial in for public comment. They they want to make a public comment. So what I'm going to ask the committee to do is maybe go and use this time to go and gather your lunch, and we'll have a working lunch. And I will ask members of the public not to interpret the fact that members of the committee are eating as any sign of disrespect, but as a sign of content abundance. So I will ask that everyone please be back at 2 o'clock. I do want to just share. Okay, I'll share it when we come back, because I wanted to put up the address to send in written comments. I'll do that when we come back. I know not everyone likes to give oral comment, so we do have the opportunity for written comment as well. So everyone, if they could please be back at 2 o'clock, and we'll be on break now.